from Microbe TV. This is TWiEVO, This Week in Evolution, episode number 76, recorded on March 21st, 2022. I'm Vincent Racaniello, and joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Greetings from LD Lab Studios. We're up and running. Our spring season of TWiEVO is underway.
That's right. So today is first day of spring, isn't it?
Yeah, today. Yeah, that's right. 21st, the
cool
equinox. Yes.
Welcome to spring. I forgot to say, you're listening to the podcast on the biology behind what makes us tick.
Thank you. Yeah, it felt a little abbreviated there, but that's okay. We brought in the music on our live stream for the first time.
I thought we'd have some music. I haven't done that in a while. We have a bunch of people here. We got 20 people, and welcome. I would say they're hardcore because, I just posted this morning. We got, right. I know, so here we go. We got Rima, who is from Nebraska. We have Jeff, who is from Nebraska. We got Franz. I don't know where Franz is from. We have Andrew, who I know is from New Zealand.
That's right. Good to see you, Andrew. Welcome back.
We have Sean. Don't know where Sean's from. Priscilla, I believe, is from Brazil.
Hey there, Priscilla.
MK, I believe, is not too far, maybe Massachusetts or something like that. And then we have Peter from Colorado. Not too far from you, Nels, right?
Yeah, just next door in the Mountain West. Just a quick hike over the Rocky Mountains onto the from the Front Range to the west side here, where I'm.
John's from New Jersey, not too far from me.
There we go.
Yeah, that's our audience today.
Fantastic. Some last-minute podcasting here. We just put together this paper this weekend, this preprint, which I think is going to be really fun. We're going to step a little bit away from SARS-2 for a moment, but maybe we should just do a quick virus watch. Vincent, how are things running in New York?
Here's another from Asheville. New York City expat from Asheville. Cool.
Yeah.
And with a few more people here. Yes, Priscilla's from Brazil. Missing Nels. How can you miss Nels? Manuel is from Portland. Nebraska wins. Environmental Coffee House from West New York. We got Barry Brown from Manhattan.
All right.
I always like to hear where people are from. I tried doing this once on the live stream with Amy. She said, "No, I don't care where people are from. Stop it. You're wasting my time."
I enjoy it, I have to say. It's really wild that
I like to see where people are from. Aida, where are you from? I couldn't guess. Alfonso's from Toronto. I just love where people are from. There's this, you can actually get a globe that goes into the software live stream. It shows where people are from as they pop into the chat. I think that I have to get that. Yeah. That's pretty cool.
Yeah, absolutely.
Anyway, we always have a great bunch here.
I agree.
On TWiEVO. All right. So what's happening in New York is that, cases went way down. Aida's from the Bronx. Yo, how's it going? The Bronx. I was there a few times. And Michael's from Los Angeles. Cool.
Welcome, Michael.
Here in New York, the cases, of course, have plummeted since the peak of Omicron. Daniel's from Mexico City. Very cool. Very cool. And Krister is from Sweden. Who has a COVID question. I'm going to leave to Nels. Franz is from Amsterdam. How cool. We got Biolution from Central Switzerland. Going to be in Switzerland in May. Going to be in Zurich. We got Eddie McGregor in Salinas. We got Richard in the UK. You don't mind this, do you, Nels?
No, I can't get enough of it, to be honest with you. Yeah, we can just do this for the whole hour.
Well, and then, they're here to hear us. So, someone wanted to know what's going on. Oh, here we have Finland. Wow. Fantastic. Compare. John is in the Peak District. I don't know what the Peak District is. Do you?
No.
I'm showing my ignorance. Yes, I think it's rather cool to see how worldwide this community is. Absolutely.
Absolutely.
Viruses affect the world. They don't know borders, right, Nels?
That's Of course. Yep. And that's what we've been up against for a while. Somewhat, somehow I think it's interesting. We've all kind of retreated in a lot of cases indoors and kind of almost into semi-quarantine in some cases. But this sort of these new ways of connecting globally with sort of some of the shared experience it's really a silver lining or good opportunity coming from this.
Anyway, so, to answer your question, New York cases went way down. They haven't gone down to zero. There's kind of a little rise at the end here. I mean, we're going to rise, just a plateau, right? It hasn't gone to zero. And I think that as the weather gets better and people move around, they're moving virus with them. So, but here, no more mask mandates in the city, and only the transportation hubs you have to do that masking. Here, John is the first national park in the UK. Oh, Peak District. I didn't know that. Did you know that, Nels?
No, we're learning here as we go. Yeah.
Excellent.
Very cool.
Thank you, John.
Welcome, John.
Thank you. How about in Utah? What's going on there?
Yeah, so things have cooled off here too. I think, in the state, the numbers have been about 100 cases a day for the last week or so. We're all kind of, wondering what the next step is. I heard it's a little bit dated now, maybe to end January, but I heard Trevor Bedford, who's at the Fred Hutch in Seattle, who's done a lot with some of the genomic epidemiology and related work, and at mystrain.org. And he was kind of, speculating that there'd be a long tail on Omicron. And this is, we've been talking about a little bit on TWiEVO too, this sort of, I've heard it called a subvariant, the BA.2.
Yeah.
And I'm, and so, and of course that's in the news headlines right now, is just wait as the next shoe drops on the US. Not my cup of tea or experience, but if you kind of look at the curves that have happened in Europe where we're seeing those upticks, in some cases BA.2 got into the mix a lot earlier than it has here. And so I think I heard we're at 30% or something like that. And so, I don't know if that maybe puts us closer to Trevor's idea that BA.1 had such a sort of, successful run in the US. Does that take a little bit of the sting out of BA.2? I think that's an open question, but that would be sort of a best case scenario. Yeah.
We have Christopher from Scotland. How about that?
Cool.
And Richard said Omicron is massive in the UK right now.
Right.
Yeah. So on the train in this morning, now as I was reading two articles, one by Eugene Koonin, where he basically does modeling to look at the receptor binding domain. And he says, there's not much more change that can happen here. He said, it's a boring conclusion. He actually has in the title of the paper is the conclusion is boring. But, and then on the other hand, there was another opinion piece saying the number of variants are infinite.
Yeah. Well, so the truth is probably somewhere in between. So I think the number of variants are infinite. The ones that work are limited.
That's right.
And that's right. And so that's where Eugene's coming at it. But, oh man. Yeah.
No, I actually thought you should probably be in on the Koonin discussion because it's all modeling, right?
Yep.
And I'm not sure that I could do a good job on that, but it's an MBio paper that just came out. And I had actually been asked to review it, but I never responded. But I should have, because he was the kind of thing where he's already got the reviews done,
I see.
The title is extremely, provocative, right? The boring conclusion is that nothing much more is going to happen, at least with changes around the receptor binding domain. That's his point, which is not the whole story, of course.
Yeah, that's right. And a lot of folks are kind of, as they're thinking about the next steps, are sort of inferring that the immune evasion is kind of the game now for SARS-2, and how that all adds up in the sort of combination of variation that could get around recognition, increasingly less naive human population. That's definitely feels like a kind of an uncontrolled experiment that's running even as we speak.
Yeah, let me show you this article because you got to see the title. It's so funny. Nope, that's new. There it is. Epistasis at the SARS-CoV-2 receptor binding domain interface and the propitiously boring implications for vaccine escape. Don't you love it?
I see. So yeah,
propitiously boring.
Classic Eugene Kuhn in there.
Yeah,
I think the last time I saw him was when we were together at this is now 2+ years ago, at that meeting in Germany, the giant virus meeting.
That's right.
Yeah.
But
well, Vincent, we're taking a break from SARS-2 evolution today to kind of jump back into the game here with sort of exploring all of the vast kind of studies of evolution out there, and some really interest. I mean, there's just been paper after paper, preprint after preprint that have just been sort of waiting in the wings here as we've been distracted, or not distracted, but just sort of engrossed, maybe is a better way of saying it, with SARS-2 evolution for obvious reasons. But so today's paper, or preprint, I should say, is titled "Single Mutation Makes E. coli an Insect Mutualist." So we're not completely out of the microbial world here. We're moving from viruses to bacteria. And we're also switching hosts from humans, in this case, to insects, and thinking about the evolution of mutualism, or of symbiosis here. And so maybe before we dive in to the preprint, which I think is just really fun and kind of inspiring.
It's the kind of work you can do with E. coli,
Yeah, so it's so fast, the tools, I think, and it's just colliding that with this complex evolutionary biology is really, really exciting. But let's pause for a moment and highlight the authors here. So it's the first author is Ryuichi Koga, and the senior author is Takema Fukatsu. This is coming from the University of Tokyo, Japan, Department of Biological Sciences, and also an institute, the Bioproduction Research Institute at the National Institute of Advanced Industrial Science and Technology in Tsukuba, Japan. There's about maybe a dozen authors, and I won't sort of torture everyone with trying to pronounce all of the names here, but and available on bioRxiv. So preprint here. I'm guessing this is in the middle of peer review at the moment. We'll do a little podcast peer review, as we like to do with the preprint. But don't be surprised if this. I would guess in a few months, or after some revisions, that we'll see this paper probably in one of the magazines, one of the sort of fancier journals. Yeah, has that kind of it's a tight story. It's kind of organized to kind of get that one exciting point across. And so, but before we get into the details, let's back up and just sort of think about mutualism, symbiosis, these things. So turns out, we ourselves are the beneficiaries or victims, depending on how you want to think about it from a philosophical standpoint, of the symbiosis. And of course, I'm talking about a really ancient event, the acquisition of what became our mitochondria, the endosymbionts in all of our cells, and that we share with other eukaryotes for a billion plus years or something like this. So it turns out there's a really rich.
So is that is that also a mutualism, that mitochondria, or is it just so symbiosis? If I understand, is just two things living in proximity, right?
Yeah, and so there's also in this is a whole kind of lexicon here, technical lexicon. I probably won't get every detail, but so obligate symbiosis, where both parties or one or the other party depends on the other. And so that's usually, this is more than sort of a passing association. It's really permanent if the species or the organisms are going to survive. This is different than parasitism, where there it's, if we think about, we can think about viruses as parasites in some cases, or, leeches or something like that, where the parasite comes, gains some energy from a host, perhaps reproduces, and then moves on to another host, but it's sort of a transient association. Could be long-term as well. But there's sort of a clear, or a more clear situation here in terms of one party doing better, the other doing worse. and sort of, this sense of some exploitation, the sort of more colloquial definition of parasitism. I think one way of describing mutualism or symbiosis is actually to use the word addiction. Now, you have these sort of biological addictions where you come to depend on something that's provided by the other, and then when you pull that away, you get in trouble. Things go off the rails here. And that's certainly the case here with this obligate symbiont that we'll be talking about. And so, as I was saying, like, this turns out to just be all across the board in insect species. You have all these incredible associations with cicada bugs. We'll be talking about stink bugs today. And sometimes the biology here can get like super complicated, and not always for reasons that make sense. It just sort of has become complicated as these associations have gone on for a long time. A lot of the insects actually end up with these specialized organs that house the bacteria that provide a location for them. And that helps, especially in vertical transmission. So as the bacteria are passed from mother to offspring, you kind of set up the plumbing of the GI tract basically in order to promote those kind of associations. I mean, it looks like in some cases it looks like an engineer was in there, or like a remodeler or something. But this is all kind of can be explained sort of by classic Darwinian principles of mutation and selection somehow. But it's all this complex biology. So that, I think, really underlines why this is a fascinating question. How is it that two separate free living species might become associated with each other and become addicted to each other? And so the addiction here for the host is oftentimes the bacteria is providing essential nutrients, things like amino acids or other nutrients. And so, here we've got a more formal definition here, Vincent. Maybe walk us through.
This is a nice little picture here. So we have mutualism. So we have a flower and a bee. The flower gets pollinated, the bee gets nectar, right? That's a mutualism. Arrows going back and forth. Both groups are happy. Commensalism, one way, the barnacle and the whale. The whale is unaffected, but the barnacle finds food, right? So the whale could care less, I suppose.
Probably cares a little bit, but it's all kind of on this spectrum, right?
It's very hard because it's also from our viewpoint, right?
Exactly. Yeah.
And then we have parasitism. We have parasitism where the tick gets a blood meal, right, and the cat gets a skin infection. So parasitism, and that's very, viruses are parasites. So they take from us, and I don't know. In some cases, viruses may be beneficial in some way, right, Nels?
Oh, absolutely. Yeah. And so actually there's some great examples of symbiotic viruses, again, in insects, actually. So like parasitic wasps will, when they deposit their young into like a caterpillar larvae, they'll also co-inject, in addition to the eggs, virus particles, which then turn off the immune system of like the caterpillar and allows their eggs to grow without as much of an immune response. So absolutely. We see kind of all shapes and forms of this. In some ways, for me you and me as virologists, those are some of the stories that I'm drawn to. Some of these viruses that can change the behaviors of the hosts, all these kind of things. But I have to say, like the bacteria, and we'll see this in a few minutes here, the biology is absolutely beautiful, and the systems can be kind of manipulated. And that's maybe one of the real breakthroughs here, is actually bringing E. coli into the mix. And in this sort of experimental evolution, as the title suggests, seeing this step towards mutualism in one mutational step, which is really sort of surprising. It's not this really specialized, the actual symbiont, we'll talk about this, and how they quote unquote cure the stink bugs. But it's, this is really incredible biology. We talked a little bit about this, Vincent, back on TWiEVO. This is, I looked it up, episode 25, in the company of Cnidarians. And this was a mutualism between dinoflagellates, these small critters now are in the ocean, and Cnidarians, so relatives, sea anemones, relatives of I think these are like reef dwellers, the corals, etc. And the study that we looked at actually considered the host side. So how is it that the host, the genes that it encodes that might promote, in this case, the sort of the association with the dinoflagellates. Here, we're going to switch completely to the symbiont side, to the bacterial side, and follow along as the authors do some really clever manipulations to like actually set up a new symbiotic situation experimentally. And so, as I kind of was hinting at, so the creature here we're talking about, the authors uses the brown-winged green stink bug.
Stink is not fair because it maybe doesn't stink to the bug, right?
Absolutely. And there is this old saying, like, the thing that you pick to study as a researcher says something about you. So I don't know what that says about the authors here, but they seem to be fans of stink bugs. I'm a fan as well, so count me in. And in fact, there's some gorgeous pictures of these stink bugs. We'll have to show them here. The way the symbiosis works is, so like a lot of insects, the stink bugs have this sort of specialized part of their GI tract. I think they call them these crypts, or these sort of cavities. And this is a spot where the regular symbiotic bacteria, which is in the genus Pantonia, Pantoa, resides. And Pantoa, so it's a distant, like really distant cousin of E. coli, but in the kind of same family, roughly speaking, which kind of gave them, I think, the courage to try this association with E. coli. But the stink bugs need Pantoa. They need this bacteria. They pass it vertically. So mothers, as they're laying eggs, inoculate or this bacteria gets added onto the egg shells. And then the nymphs, as they hatch, start foraging around. And then they ingest this bacteria, the Pantoa. And then that and that doesn't happen 100% of the time, but it's necessary. They are obligated to get a hold of this to actually develop, to emerge properly, and to develop. And so only, even under at least in lab conditions, they've kept these stink bugs, I think, for like a decade or more in the lab. Oh wow, here we go. Yeah, so here it is, the brown-winged stink bug.
Yeah, Plautia stali.
Yep.
And now I have to say, I know that species because there is a picornavirus that infects it, and that has been studied quite extensively. Wow. Yeah.
Oh, that's cool.
How about that?
I love it. Yeah, distant cousin of polio as well. Maybe there's a virology story in there somewhere. Who knows?
Always. Always.
Yeah,
right?
There must be. So anyway, as these guys are going along their business, hatching and growing, they associate with this new bacteria, and only about 70% of them make it. And so it turns out you can sterilize the eggs. You can get rid of the Pantoa bacteria. I think I was glancing at the methods section, and they treat the eggshells with formaldehyde and then rinse it with water. And the larvae, the nymphs, hatch just fine, but they all die without that bacteria. And so that illustrates, so this is like important. And in fact, I don't know, can you pull up do you have the preprint up, Vincent?
I do.
Okay, so let's look at figure 1, because it's just kind of, a picture is so much better than 1,000 words or whatever.
I got to find it here. Figure 1 you would like, right?
Mm-hmm.
This is the one, oh, wait. Figure 3. Oh, sorry. The figures follow the legends. Okay, here we go.
Yeah, exactly. Yep.
So this is the one with the nice colors. Is that what you're talking about here?
Yep.
Oh, yes. Here we go. Let me get this into my field of vision here. Oh, yes. This is very cool. Very cool.
Yep.
Here you go.
We got to show it. Yeah, here we go. Yeah. There they are. So that's so
beautiful, aren't they? Aren't they beautiful?
Yeah, I agree. Incredible. So if they gain that bacteria, they're symbiont infected. These are adult females. You can see that beautiful green color that's sort of looks like just a healthy stink bug for someone who isn't really a stink bugologist. Now, and if you sterilize the eggs, as we were just describing, they don't hatch at all.
That's this guy here, right? Oh, no.
Well, he's become E. coli, he or she has become E. coli infected.
Yeah.
And so it's alive. It's actually, it's made it through some development, but it doesn't have that great luxurious green coat. Yeah. And it looks like it's missing an antenna. This is not a healthy stink bug. You can even think you can tell that even if you're not an entomologist. It's not a happy critter. And so that really, I think, just dramatically illustrates the addiction. They need this bacteria, this Pantoa bacteria, below their midguts. And so here we can see they've dissected out the digestive tract, and you go from the midgut over to the symbiotic organ that they've highlighted. It's in yellow. That's where the bacteria, the Pantoa, are hanging out, providing something. The authors don't go into the details. I think there's some literature here, so there's something known about the biology here. What is it that the bacteria are providing? But then what they really focus on is that in panel I there. So this is the guy that's been, so they've basically taken away the natural bacteria, and then they provide back E. coli. They don't actually put it on they don't try to treat the eggs with it. What they do is soak cotton pads with the E. coli that they're using. And these are like lab strains of E. coli, by the way. And there's some cool details here in a minute. And then those nymphs, as they're sort of foraging around, to get a little bit of that E. coli, but it's nowhere close. So you don't see like the coloration even in the symbiotic organ, the crypts, which is near the hindgut, near the anus. Not much going on there. Although if we scroll down a little bit, we can see in pink, there is a little bit of E. coli there. And that's so the panel C and J show that there's even the E. coli, it's hanging out there. There's not as much of it. The crypts aren't as that changes the morphology. And then we're zooming in below that in panels D and K. And so you can see now this is why they call it crypts. It has this sort of tortuous kind of back and forth so that you can really accumulate these bacteria at high density. This is a specialized organ that sort of evolved over who knows how long. There might be some estimates on that out there to accommodate the bacteria that and sort of the symbiosis has become more and more ingrained over time. So that kind of sets it up, so basically, you really need this bacteria. If you have E. coli, you're not doing so well, but you're kind of eking along. And here's where this is important. So this is like why this kind of might work is they say that, not 70% emerge and grow, but something like 5 and 10% can just sort of muddle through having the E. coli provided to them. And so for an experimentalist, this is gold, because you can now imagine using experimental evolution. So basically setting up this association experimentally, allowing it to unfold a little bit for that, 5 or 10% to come along, selecting on some of the population, saving a little bit of the bacteria, using some of this bacteria for a new infection or association, and then sort of just continuing on these serial passages, as we sometimes describe it. And then seeing, in this case, could the bacteria adapt? Could there be a mutation that sort of gets it closer? Do the bugs do better based on the traits that you select for, or the phenotype you select for? And so they chose 2 ways to do this. One simple way was the color. So a green stink bug is a happy stink bug. and so I think, and so basically, as they're setting up these infections, pick the greenest ones that emerge from the crew, and then collect the bacteria from those ones as they grow up a little bit. And then the other trait that they selected on was just how fast they grow. So the first ones to emerge, the idea being that they're somehow doing a little better than the brothers and sisters that are in the clutch emerging from those eggs. And so by selecting on that, you hope to that there's something about that bacteria so there's a genetic change. That's sort of the home run here, that there's a change that then makes it better. And then you kind of select over time. And so it works. And by the way, it's not like a slam dunk here, Vincent. So they say that they tried a couple other species of bacteria. I think B. subtilis, which is another Bacillus subtilis, which is a well-known to microbiologists, well-studied, really fascinating bacteria. And then Burkholdia, I'm not pronouncing it right, Burkholdia, which I think is a pathogenic bacteria.
Right.
And neither of those rescue. So it might be that the E. coli is sort of close enough to that Pantoa. There's something about the metabolism or just the biology that they're kind of like not pre-adapted, but they're just a little bit closer, and it might be easier to associate or provide something to sort of get a little bit closer to that obligate symbiosis.
It's so E. coli and Pantoia are both Enterobacteriaceae.
That's right.
Whereas subtilis and Burkholder is not. So that probably is part of it. Being evolutionary distance, right?
Yep. Yeah, no, it's really interesting. And bacteria are obviously some of them, a lot of difference in morphology, but a lot of bacteria kind of look the same to us too. And so not like recognizing that there's vastly different biology here, and they've kind of connected it close enough. And this actually raises an interesting point. So they call this process, or, what they're hoping for is what they call an incipient symbiosis, basically a restoration of a symbiosis. And so it's actually, this is important, and maybe to step back and kind of put this into a little bit of perspective. What are we really looking at here? So we're not creating a brand new symbiosis out of whole cloth, or a brand new mutualism out of, it's not starting from scratch. Obviously these stink bugs have these, as we just were peeking at, have this really kind of specialized crypt morphology in their kind of end of their hindguts, near their hindgut. And so a lot of evolution has already happened. And it's interesting to note, though, so that for this species, and this might be part of the reason they chose the experimental system, is it's known that it's not the most stable symbiosis, actually. So there are times where the Pantoa might get kicked out, another Pantoa might come in just from natural associations. And this is kind of an important, I think, not just a detail, but sort of an important thing to think about with a lot of these complex biological events. I think certainly I kind of think that, these are stable systems, that, we look at it over, we kind of think about it over, our lifetimes or something like that, over evolutionary time. There's all of this, there's a lot of change for something that appears to stay the same. And so I think that, that's a really interesting question, actually, is how is it that a replacement event could happen? And how necessary has that been over the course of the symbiosis to sort of refresh the symbionts, which will become more and more inbred the longer they, stay associated, and the genomes change really drastically. We'll talk about that a little bit. And in fact, some of these symbioses, I think when we were talking about those cnidarians back on TWiEVO 25, some of those, they can get turned over like by the week or by the day in some cases, with the tides even, in some of these systems. And so you have the whole range from something that is super stable like mitochondria, all the way to something that might be there for a few hours and then flushed out with a new mutualist coming in really quickly.
Yeah, so some of these interactions, you're familiar with the bobtail squids, right? They have bacteria that emit light so that they get cloaked at night against the moon, and other predators can't see them, right? So they glow. And in the daytime, they burrow into the sand, and the bacteria leave. And they come back at dusk, a whole new crop. And so the opportunity would be there for new symbionts, mutualists, right?
Yeah, exactly. Yeah, that's right. And so this one, I think, my sense is this is a little more stable, but it's not completely stable.
Yeah,
it has this turnover. And that might, it's kind of finding this, almost this Goldilocks of things being just right for the experiments to work. But if it's, right. And I think looks
Makes sense.
They've kind of found that. So
Nels, let me ask you. So they, at some point, the emergence of adults went up, right? They got more adults, like it was generation 7, I believe, in the color selection. And so did they, after that, were they able to let them just grow on their own, or did they always have to do the isolation of V. coli and then put it onto a new one? Do you remember?
I do. So when they continue doing it, it's a big experiment. So they do those 2 selections that we mentioned, either on growth or color.
Right.
And then I think they do like 12 replicates populations for color and 11 replicate populations.
It took 2 years, right?
Growth. Took them 2 years. Yeah. They said under best case scenario, it might've taken a year just with the generation time. But remember, these are sick stink bugs. And so, just kind of nursing them along adds a lot of time. Sometimes they go extinct, they fail. Yeah. And they have to and then they go back. So each time as they're infecting the bugs and then allowing them to grow, they'll save part of the collection of E. coli that they get, and then the other part they'll use for another infection. And that actually saved them on a couple of occasions, because they can go back to the freezer. They have sort of a fossil record, in a sense, over the course of the experiment. It's not like an old fossil here, a 2-year-old fossil, but then they can kind of restart the experiment along the way. And so, you're exactly right, Vincent. So in one of I think the growth selection by generation 7, they noticed an uptick every time they did it, and then they kept going. And then even, I think it was even earlier in one of the color selected ones by like the second generation of the bugs, of the exposure of the bugs, they start to see an increase. And then that continued through the whole course of the experiment. I think in the end they did 12 generations of bugs. So 12 infections with the bacteria, but that wasn't 12 for 12. It was 1 out of 12 of the color selection and 1 out of 11 in the growth selection. And so that kind of like illustrates how this is just barely they're barely hanging on here by their stink bug fingernails in order to survive this. They're missing their regular symbiont. The Pantoa species is missing here. And so this is like a bad match at the beginning, very rarely. And does it work? And in fact, they had to stack the deck in their favor. So the bacteria they chose, the E. coli, now they're taking advantage of some of the vast knowledge that we have from E. coli. So they used this mutator strain. And so it's missing a gene called mut-S, which is involved in DNA repair. And by taking that away, the mutation rate in those E. coli is up 100-fold. And so the bacteria are generating 100 times the mutations they would across the genome. And that turns out it was probably key to this working in that 1 out of 12 or 1 out of 11 times, because they do another control.
Yeah.
or something like a control. They do another parallel set of experiments with E. coli that are deleted for a different gene they have a normal mutation rate basically, and those guys all go extinct. They can't keep the experiment going. And so this is a preprint, so it's freely available. Definitely worth checking out. I think it's like as we've already seen, the figures are beautiful, and the way they present it, it's just it's complicated biology, but it's kind of cleanly delivered. And so and so they show these, like the 12 lines, they have a heat map where they're able to see that when they emerge or what the color is based on their metrics, and you can see one line goes all the way through, the other 12 lines go dark, they go extinct. And it turns out it's both the green color. So if the greener the color, the faster the emergence, the more the growth. These things correlate, which is not surprising. And the E. coli is changing, so they can still, it's a free living bug, right? And it still is, even over the course of the experiment. They can grow it on a plate, but the morphology of the E. coli changes. This is really interesting. Different colors and sort of textures as the colonies grow up on the plate. So of course they're like curious, and hopefully we all are, like, what is it that happened over this like really short course of a few months basically, how did the E. coli change that might have given it this sort of property so that it can help to nourish the stink bugs.
One of the traits that the new symbiotic E. coli has: slower growth rate, smaller cell size, unstable cell shape, loss of flagellar motility. I find that really interesting. Yeah. Doesn't have to move around in the world anymore, right?
That's right. Yeah, no, and this is like really fascinating. And it kind of, this addiction goes in both directions in a lot of these cases, right? So as the E. coli loses starts to let go of some of its features, like the flagella, it starts to depend on those mother stink bugs when it's laying its eggs for that bacteria to then be on the surface of the egg so that the nymph will eat it. So that's like, instead of having a tail to swim through the water, you now depend on the mother stink bug to basically feed you to the offspring so that you can find the next crypt where and that's a whole nother like fascinating part of this biology, like the shape in the crypt, but also what are the E. coli, like what's keeping them alive, or the Pantoa, what's the host providing them, sort of both sides of the addiction here. Okay, so to start to crack that question of what's going on here, in addition to those changes in morphology, the loss of the flagella, and those kind of things, what's going on? So the first thing they do to get kind of at the molecular basis of this is RNA sequencing. So they're looking at so they harvest the bacteria, grow it up, get the RNA out of there, which is going to include the mRNA messages, sequence everything, and then compare the transcripts between the ones that have evolved or the ones that have been associated with the stink bugs versus the ones they started with, the parental E. coli, which is just a normal lab strain. And so I think they report there's actually, so this is a little bit toughy. This is a little bit tricky because it's a 100x mutation rate. Bacteria grow really fast. They have tons of generations in a few hours. There's hundreds of mutations here actually. And so they have to start doing some comparisons. And so to try to narrow it down. And I think they claim there's 200 genes that are shared genes that are downregulated, whether they did and there are two examples of some adaptation. So both the green selection and the growth selection, 200 genes shared that are turned down and 100 genes that are shared that are turned up in the independent lines. And this sort of hints at there's a lot going on here. And it's not just sort of one single. So it's going to come down to a single change, but it's a complex single change with a lot of knock-on effects. This changes the transcription program in some way. And so, and they could already start to ask, well, what are the functions of these genes? And it turns out that they're a little, not completely simple, but one of the shared features is this so-called CCR pathway, which is the carbon catabolite repression pathway. And we'll get to the details of that in a minute, but then I think I would have done this first, or maybe they did this first and then are just reporting it in reverse order. But then, not only did they sequence the RNA, which gives you all of the transcripts, they sequenced the DNA. Again, hundreds of genes, but using the fact that they had two independent events. And this is why I sort of on TWiEVO drone out about the power of convergent evolution, independent evolution. You have two things that end up in the same place and you can compare them. Now you can make a much stronger conclusion. You go from hundreds of mutations to the ones that are shared between those two independent events. And now that narrows you down, I think, to 10 candidate genes. And then they are all in the same pathway, the CCR pathway, carbon catabolite repression. So let's talk about that, and then we'll talk about the killer experiment, and then get into the discussion here. So, so this pathway is involved. So usually, especially if you're a lab E. coli, like these ones are, and you grow together in nutrient-rich broth, you have all of your carbon source is generally glucose. You have all of the sugar you need, and so under this condition, it's sort of full steam ahead. The bacteria can replicate really fast. However, if they exhaust that media and grow to high density, then they'll trip they'll sense the lack of glucose and then trip this pathway, the CCR pathway, to start to utilize other carbon sources that might not be as efficient to get energy, to make ATP, but it's doable. And so what's interesting here is it looks like that pathway has been lost in the E. coli. And so, and it's not just that they're living in this, so the authors say this, they're living in the crypts and that might be at high density. And so if you lose that pathway, then you're not wasting the energy to try to use other carbon sources. You're just okay, I'm here. And, there's a little hand waving. But whatever the explanation is, and we'll spend a little time maybe digging into this in a few minutes here. But what they could do is, given that they've narrowed it to this pathway, now they could do the clean experiment. That as an experimentalist, you just you kind of crave this. For me, this is the pin. You have maybe a few experiments in your lifetime where you just test something and it works, and it's just this really cool outcome. This feels like one of those events. You've got the CCR pathway, so you say, okay, E. coli, this is easy. We can make a simple point mutation in one of the genes necessary for that pathway, or just delete one of the genes. So they do both of those things. And then we can take that E. coli that doesn't have the mutator phenotype. It's normal E. coli strain with those precise, single genetic changes. Let's put that on the cotton pads for the cotton balls for the nymphs and see how they do. And this clean E. coli with those single mutations it worked. It had the same outcome as the ones that were evolved. So they've narrowed it. It's a really good strong evidence that there is something really important in that step to becoming symbionts, of losing the activity of the CCR pathway.
I mean, basically, there are two changes in their selected bacteria. One is it's a introduction of a translation terminator, right? and the other is a leucine to proline, a single amino acid change. And as you said, when they put that single amino acid change in or the other, so that's where the title comes from. One amino acid change, because the other, translational suppressor is not as sexy, right?
That's right. Exactly. Yeah. And I think we have to look at these stink bugs, Vincent. So this is figure 4. I don't know if you've still got it up. Yeah.
I'll pull it up. Yeah.
And,
Nice.
it's like, and I don't remember how big the E. coli genome is to begin with, but of those, like, you can make thousands, hundreds of thousands probably, hundreds of thousands at least of single amino acid changes, like across all of the genes. So how do you know which one to make? And that's where the experimental evolution gave them that prediction, and it worked. So in panel C here, these are the stink bugs that were rescued. So we saw the like super brown one at the beginning. And so if we look here and going across from left to right in panel C, so they just knock out this single gene. And then here they make this single change of a nucleotide that, as Vincent was saying, changes a single amino acid. And there you see that green bug hanging out right next to it. Yeah. And now, so if we go to the next two, though, are actually that's the bacteria with all kinds of changes that they got out of their experiment. And so
these two here, right?
Correct. Yeah. And so I think these are the ones that they were going for the growth. And they actually look a little better. Like they've got antenna now. I don't know if that's in a leg or two. I don't know if that's missing and so they're a little thin on the details. So we can see above this, the emergence rate. And so when we look at panels B or in kind of orange there, so the delta, so the first one on the left, let's go left to right. And the A, this is the emergence of the bugs that are given just the E. coli that hasn't been just the parent at the beginning of the experiment. It's just sort of muddling along. And then by making those two changes, either you knock out this gene or change one amino acid, you can see that bump up, but it's not up to that level C in blue or green. And so green is the original symbiont. But even the ones they did for that 12 generations, you can see the emergence rate is really,
it's pretty good. Yeah.
Yeah. It's right back in the game.
Yeah.
So that's kind of it. So that's it's sort of like this,
single amino acid. It's amazing.
Yeah. And so the fact that you could be able to narrow it down so quickly, I think is pretty remarkable. This leaves
put this in perspective, Omicron has 34 amino acid changes and interesting phenotypes, and it's going to be a long time before anyone sorts that out. It's a lot. For this E. coli genome, which millions of base pairs, right?
Yep, that's right.
It's for this to be the home run is amazing. It's absolutely amazing, as Nels said.
I can't get enough of it. And sort of illustrates how close, so some flexibility for the ability to pick up. You don't need, you also don't need this massively specialized Pantoa that's been in other stink bugs for a really long time. There could be environmental samples out there with even one mutational step that could be a viable replacement. And so this opens like a million more questions, which is, I think, the best studies do this, right? It kind of opens up an entire kind of new collection of experiments that you could imagine doing. So one of the questions, which I would guess they're continuing the work now, is what happens next? So they went 12 generations. What if you go 100 generations? That might take 5 years or something like that. But to then follow how the genome changes in this sort of at the beginning steps of a new symbiosis. This is a fascinating question. And we kind of know what happens, like just from snapshots from long into these associations. And this is from other insects. This is even, if we wanted to take it all the way to mitochondria, the genomes get like eroded. They lose, like we already saw in this first pass, right? You lose the genes that are involved in this CCP pathway, but then very quickly other things start to kind of go to the wayside. It's a pathway. So there's like dozens of genes involved here. Some of those like 300 or 200 upregulated, 100 downregulated, or it might be the reverse of that. So if you don't need those any longer, there's not selection on those bacteria to keep them around. They're not free living anymore. And so how does that process unfold? How does the genome become streamlined or degrade? And there, we imagine that there's some reasons for this to happen. If you can replicate a little, so there's not only are we worried about the stink bugs, is selection worried about the stink bugs emerging and doing well as they're competing against other bugs, including their own sort of brothers and sisters. The bacteria is competing too. And so the bacteria that can replicate a little faster, and that might be because they've gotten rid of this now genomic dead weight, so to speak, that they don't need now that they're establishing this new relationship, they're becoming addicted, they very quickly start to lose big chunks of their genome that aren't needed.
So
again,
what is that?
A random process.
Is that by like recombination, which always happens, right?
Absolutely. Recombination, insertion, deletion, depending on, if there's phage in there and how integration is happening, like all of these kinds of things. But we honestly don't know the details of that. We usually just see, okay, here's a really degraded genome, or you also get this like complexification. There might be the bacteria split into different populations. They start to feed each other. The cicada bugs have taught us a lot about this. Yeah.
So they can look at that because there are recombination defective E. coli that they could say, okay, let's put these in the stink bugs and see now if we get the loss or not, right?
Exactly. So I think that's to me, the really fun here is of using a model system like E. coli that has, exactly as you're saying, Vincent, that has all of these tools. You can just quickly imagine ways of dissecting out the process and say what works and what doesn't work. If we give you that jump start, we just make that one amino acid change in the one gene that then starts it along the pathway here. And then you can just go to the races. Yeah.
Now, one thing here, this is lovely, but remember, most E. coli live in intestines already, so they're probably really close to symbiosis to begin with, right?
That's right. Yeah. And that's right. So this worked for E. coli. It didn't work for two other, for B. subtilis and for
which are not, I mean, B. subtilis is a soil dwelling bacterium, right? So it's got to live out in the world tough, all on its own. E. coli in the cozy intestines is probably close to being a symbiont to begin with. So, I mean, it still is a very cool experiment, but it would be even, and probably that's the only way it would ever work, because maybe you could never get B. subtilis to do this, right?
I don't think you could get B. subtilis to do this, but stay tuned. There's more work coming out. I think the field's just sort of poised for kind of an explosion along these lines. I know of another case, a totally different insect, totally different symbiont, but I think the bacteria comes from an environmental sample and then does this sort of crossover of kind of independent example. And I think the kind of overall surprise here, or maybe this is kind of breaking down dogma that symbiosis is rare or really difficult to achieve. Here, I think what we're starting to learn is that you can just do this, like it's easier than it looks, which is surprising. And we haven't dug in yet. And the authors don't either. They kind of, I think they're excited about this result, and I don't blame them. But we don't get much of the background here, or even some insight into okay, so what is the bacteria actually supplying here, or what is so right? Because we know that the CCP pathway, carbon catabolite pathway, is kind of knocked out. But like, what does that mean? Not just for the bacteria kind of changing their physiology or their metabolism. How are they differently influencing the metabolism of the host, of the insect? So how is it that the bugs are turning green? So if you give them the parental E. coli, they're still brown and kind of sick and muddling through. But now, after this course of experimental evolution, or that single mutation, they actually take on this green color. So what is it that is there intermediate nutrient that gets shunted off or something like this by shutting off CCP? Why are they green? Why do they look so healthy? And a corollary to that, why do they grow? Like, how are they growing better? Is there, again, some sort of nutritional product here from that one mutation that becomes available? It's both fascinating and I think, probably a little bit tricky to figure out, but that would be some more next steps.
Many people have spent their careers working on these kinds of symbioses. Nancy Moran, for example, has worked on these for many years. There's some highly reduced bacteria, right, where the genomes are 120,000 base pairs of DNA, which they stay inside their host all the time. The pill bugs that roll up, they're another common host that are used. So it's really intriguing to understand how this works, as you said, not just how it works, but also how it arises to begin with. And this paper is good on that because I think it's not easy to get at that point.
Not easy to get that. You also get the sense that they're really like, they love the sort of the chase, the experimental evolution here, right? Like that they got it to work, and then they were able to pin it down to a single change. Like that was like, they're like, look at this, everyone, this is super cool. And I think they're right. But yeah, then now it opens up all these other questions, both like the genetic process, just as you're pointing out, Vincent.
Yeah.
So like, how is recombination going to influence sort of the steps ahead, but all the way to the physiology, what's going. And this is where from Nancy Moran's lab, I'm thinking about John McCutcheon's lab, so several others who are doing some incredible work here, and taking this all the way. So, we see a little bit of the microscopy in this case, like the fluorescent bacteria hanging out in the crypts. But some of the work that's emerging here is all the way to EM, and you can see the, the host membranes and how the bacteria is associating with things like endoplasmic reticulum and these like really kind of complicated cell biological structures. So yeah, some of the work from Nancy Moran that you're describing, where it could be as many as like a dozen different kind of reduced genomes, and you put together the metabolic pathways, and it's like number one gives you these three genes, number two gives you these three genes, like, and it's just cool. Like, some way of exchanging all of these proteins or associating it so that you're running metabolism between multiple independent parties. It just gets super wild. Yeah.
So I have to say, we have a lot of stink bugs in our house for some reason. They're brown. Usually you find them dead. So I think they wander in and then they dehydrate, right? Because there's not much water. But I'll be downstairs in my office and they just drop on me. They've dropped on my shoulder. One night I was podcasting and one dropped right on the microphone. It's just, and they don't do anything. They just drop and then they sit there. It's very funny. Maybe they have a different bacterium.
You got to have a petri dish of E. coli with that.
Feed them
mutation in the CRP gene. And then
should we do some questions from our audience here, Nels? We got a bunch. First of all, Jeff wants to know: is that a fish on the whiteboard behind you?
It is. Yeah, guilty as charged. That's it's my attempt at a zebrafish with some work we're doing, not on a symbiotic event, but on a pathogen transmission event that we're thinking about. Yeah.
It's related to the picornavirus of zebrafish that you discovered, right?
Exactly. Yep.
Andrew, can we tell if this symbiotic system started as a parthenogenesis, or could it have been a sort of bargaining process?
Oof, great question. I mean, this gets at, the host. So we kind of been glossing over the details of the reproductive biology here. And so, for one thing, all of the, bugs that matter here are the females, right? The ones that are laying eggs and then vertically transmitting this, where the males kind of take a back seat. The males need that bacteria. So as they hatch and they're all nymphs, they need to get it too. But this is fascinating, right? So the E. coli or the Pantoa that the males have, that's a dead end. In most cases, because they're not laying the eggs, they're not passing it vertically from the crypts. They still need it. And so how sexual reproduction then gets tied up in these symbiotic events becomes pretty fascinating. And the evolution that can happen here, because it's asymmetric, this can kind of go forward differently, or sort of the evolutionary outcomes are different for the sex that you choose. And so you could absolutely imagine like that there's some complex, like, so the females become parthenogenetic, they kind of cut the males completely out of the equation here. Or you can just keep like normal sexual reproduction, where half of the offspring basically becomes a genetic dead end, whereas the male, and by the way, that's sort of how mitochondria works, right? All of the mitochondria that we each have is from our mothers, not from our fathers. As it gets deposited in the eggs, or is in the cells that form the eggs and then become the embryo. So
Krister has a SARS-CoV-2 question. I think I will wait to get through some of these. I'll come back to you, Krister. Okay.
And to get to Andrew, to get your second point too, the bargaining process. So I think of it more as like, I think that's like a beautiful way of framing it, bargaining, or even like, some kind of truce, or even a conflict that might emerge for who's controlling what, who's controlling the resources here. And that's another way that this biology can get complex in a hurry. Random mutations, but selection acting in different ways. Yeah, it's a really good point. Really good question.
So Jeff asks, doesn't flagella represent a very high percentage of overall energy output in bacteria? What are they doing with that energy when they drop flagella usage?
Yeah, this is a great question, Jeff. So, reproducing, dividing, if you can move all of your energy into doubling and do that quicker, that's just who's going to be around. And so that could be, I think that's certainly part of the story here, is that and the authors lift that up. They say that by sort of letting go of this expensive process and the knock-on effects, that sort of is what works. But then, with the added complexity of you have to give something on both sides for that addiction to take hold. Yeah.
In an experiment like this, how important is it to have both species being highly stable? I mean, when the bug are highly diverse genetically, I guess it's hard spotting the differences in E. coli.
Yeah, this is a good question, Franz. So, and they actually, experimentally, and this is a common theme for all experimental evolution, is you want to sort of pick what you're studying. And so they point out that in this case, they want to keep the genetic diversity of the stink bugs as they want to hold that as a stable variable. And so they basically come up with a big breeding scheme at the beginning of and I think they've been breeding these stink bugs in the lab for about a decade. But they want to have them as closely related as possible for each of the 12 generations as they're reinfecting with the E. coli and have that be what's undergoing sort of the adaptive pressure, the artificial selection that they're setting up in the laboratory. But this is a great question. So you can imagine reversing the situation. So keeping the bacteria, maybe just make that single point mutation, so that it establishes well, and then taking a bunch of stink bugs. So take, that one that fell on Vincent's microphone in his basement while he was podcasting, and it's kind of just clinging to life. Take a bunch of other ones of this species, and then give them the same E. coli, and then ask, okay, who does, are there some on the host side, are there some that are better at welcoming, these sort of or barely adapted E. coli to become symbionts? Are there some that are worse? And then what's the genetic basis of that? And then you can start to tease apart both sides. I think for this system, it becomes pretty tough. There's just not E. coli. It's kind of magical for how much we know about it, how many genetic tools there are to tease apart the biology. Whereas stink bugs, it's sort of the wilderness frontier. We don't have as many tools to be able to manipulate them. But that could be another part of that's why I think this research opens up so much possibility, is you can now see why investing in some of those tools could really pay off.
Apparently, parthenogenesis was a spell correction typo, but I'm glad that happened. A fascinating answer from Nels. That's great. Patricia.
Random evolution, random mutation and selection happening.
Well, the spell checkers do that all the time. Yeah. So Patricia wants to know if this is evolutionary biology. Yeah, absolutely.
Yeah. Patricia, not only is it evolutionary biology, but this is in hyperdrive. My friends kind of joke with me. They say, you have a podcast called This Week in Evolution, as if evolution can happen in a week. These E. coli are saying, yes, evolution can happen in a week. SARS-2 is saying evolution can happen in a week. And I think we're this is not just in these rare cases. Evolution is a lot more rapid than we kind of give it credit for.
Yeah,
absolutely. Yeah.
All right. Let me get to Krister now. He's been asking this question. Decay patterns of SARS-CoV-2 competent genomes. So apparently there's been some loss of genome sequence, right, on the order of, looks like 400 bases or so. So do you know anything about that, Nels, and what's going on there?
I haven't followed that, but that kind of is a nice echo to this case of symbiosis where there's drastic reductions in genomes. And so you can imagine this will happen too as a virus is adapting to a new host. The recent spillover that we see probably from bats to humans, or from the wet market to humans. And by the way, if you haven't already, check out a recent TWiEVO. Is this the last TWiEVO with Michael Warraby talking about
last week? Yeah,
last week, origins of some ideas on the origins of SARS-2, some evidence, and working with a small amount of evidence, but really piecing together a highly detailed picture. But anyway, so as a spillover event happens, you can imagine over the course of a few years that there's all kinds of different selection acting, including potentially for viruses to replicate faster relative to other viruses that are even in an individual host in some cases. And so if there's anything in the genome that was perhaps necessary for a previous host, but not necessary for the current host, the virus could lose that gene, or there could even be an advantage to it. So we've talked about the loss of ORF8, for example, being a theme for some spillovers from earlier, from SARS-1, and then seeing that independently in SARS-2. So yeah, absolutely. But then the question, so this happens, and then the question about the phylogenetic tracing of the decay pattern, I haven't looked at that, but that it's a great idea because you could pick up potentially independent events. And so as a virus does better because of a different mutation, say something that improves the spike protein, either for receptor recognition or immune evasion or both, then there might be a knock-on effect where something is decayed independently and you start to see that. So Krister, I haven't looked at that, but yeah, definitely worth teasing out those patterns. And, related to that, it gets complicated. So we're starting to see recombination. I think it's more evidence emerging for recent recombination in Omicron. In some cases, you see short fragments of, so between the Omicron subvariants and also with the Delta variant, there must be these co-infections where there's these swaps of information, swaps of part of the genomes of the different variants as well.
So Jeff says, we've been watching extremely fast evolution for the last 2 years now. So I would say, early on, Nels, we didn't see a lot of evidence of change in the genome, but I think the mutation rate was the same as it is now. It's just that suddenly at some point there was selection imposed, and I think the main selector was the immune response, right? And then we began to see selection. But don't confuse, I know you're not confused with mutation rate and selection, but it's easy to forget, right?
Yeah, because it all swirls together. And so we've shown some of these histograms where on the x-axis, we're stepping through time month by month, and talk about fast evolution. And then on the y-axis going forward, the number of point mutations, and it's a cloud of dots, but it's going up for the number of mutations, point mutations compared to the parent, the sort of the Wuhan 1 strain, and then you can draw a line which gives you sort of the average or the median number of mutations per month and calculate what that looks like. And then we do see these little leaps from time to time. It's still in that cloud, and how you sample the viruses will how much of a leap that is really versus just sort of an outlier. And that's where you're exactly right, Vincent. Selection's acting here. And so not only what are the mutations that are happening, but what are the ones that are sticking around? And those are probably the cases of some of these variants that we've seen, whether it was Delta, whether it was Omicron, where they have what appears like a little leap. But this is all, it's true. Mutator mutations can happen, hypermutators, but those generally, especially like in viruses, sort of don't do well. They start to kind of peter out because the genome just becomes littered with mutations, and in coronaviruses in particular, which are these massive RNA genomes relative to other viruses, there's a lot of genetic cargo that you need some fidelity to get that in through the next infection.
Well, maybe now it's time for us to do some picks, Nels.
Sounds good. Or should we hit the mailbag?
Want to do those too? Sure. Go ahead.
Yeah, got a couple of emails here. So the first paper, this is from Simon, who wrote in. I think he's talking about this episode. I don't have it at my fingertips. This
72.
Is 72. Yep.
It's about an ancient coronavirus epidemic in East Asia. Remember that?
Yeah, exactly. From David Enard's lab.
Yeah.
So Simon writes that the first paper showing the possible Asian pandemic was interesting. I was wondering if an analysis of the pandemics when European peoples arrived in the Americas would be of interest. As you know, diseases that the Europeans were largely immune to were a problem for the indigenous peoples in the Americas. Yeah, Simon, I agree 100%. There's all kinds of possibilities. I think using, I won't go really into the weeds here, but using some of these new tools to do sort of this descent analysis. So working backwards in the tree and trying to figure out in date when sort of a set of potentially adaptive mutations happen, and then correlating that with genes encoding proteins that are known to interact with viruses. And so, yeah, I think absolutely. And in that paper, in TWiEVO 72, they make the case that I think it was something like 20,000 years ago or something. There's sort of a burst of what might be adaptive mutations that could represent a pandemic of a like ancient coronavirus. And so, absolutely, I think it's very possible that you could start to see signals like that. So the question or the difficulty here becomes have the data sets. And so there's currently a lot of genomic information, relatively speaking, on Europeans. There's a lot that's probably far and away the most, although it's catching up in some circles. A lot of Asian populations, there's a lot more sequencing. Where we're really behind in a lot of cases, maybe with the exception of some of the diversity in Africa, is indigenous people in other places, Native Americans in particular, has been, there's very little data out there to do, I think, an analysis at the scale that you need to start to tease out these really, very subtle signals. So I think it's possible. Also the time scale, like I'd have to think about that a little bit, but I think it's, very likely that as the tools get better, as the data improves, or there's more data available, that there are some possibilities to kind of look at other pandemics past using some of these techniques.
Yeah, we get this question frequently, right? You hear about the European travelers bringing infectious diseases to the Americas. What about when they got there? There certainly were agents in the Americas that the Europeans were not immune to. Well, we don't hear about that. And it may be a matter of data, because a lot of Native Americans died, millions and millions, but not many Europeans went over, right? So
yeah, that's right. It's a very asymmetric situation in terms of the outcomes for those populations.
And I'm not sure how many Europeans went back to Europe to bring those, I'm not sure the nature of that one. So
yeah, no, that's right. And as we go back in our history, or kind of our known medical history, things like, certainly the plague, the Black Plague, and a few other of those pandemics are probably the ones that, at least in that time frame, what's really fascinating, right, are these ones that are sort of before recorded history. And trying to put that together with human migrations potentially. Yeah.
Yep. No, the history of infectious disease is fascinating. Unfortunately, we don't have a lot of data, right?
Really tricky. Absolutely.
Yep. I mean, you can't just rely on the written record in this case. You need biological samples, and we don't have a lot. We have another one from Anthony who writes, SARS-1 is a mosaic, and he sends a paper from Journal of Virology in 2004, Mosaic Evolution of SARS. Remember, this is SARS-1.
Oh, yeah. Wow.
So they did some genomic analysis, and they conclude that there is a mammalian-like origin for the replicase protein, an avian-like origin for matrix and nucleocapsid, and a mammalian-avian mosaic for the host-determining spike. And so this was a big deal back then because we didn't know anything. Now we know that these coronas recombine like crazy. It's part of how they make mRNAs by a recombination process. And so, they recombine as long as two can infect the cell. And Anthony writes, poultry bronchitis and feline infectious peritonitis, put enough animals together and the possibilities are endless. Yeah, there's a lot of possibilities. Yep,
agreed.
For sure.
Yep. Yeah, good point, Anthony. Thanks for pulling up that paper too. It wasn't on my radar. Very cool. All right, we should wrap up with some science picks here, Vincent. So I'll get mine going. So just saw in the last few weeks this paper that was mentioned on TWiEVO 74. So the title, this is in PNAS, Proceedings of the National Academy of Sciences. It's a commentary piece on inequality in science and the case for a new agenda. And so when we interviewed Joe Graves on TWiEVO 74, he mentioned that this was in the works and it's now published. So he's first author along with Maureen Kearney, Gilda Barabino and Shirley Malcolm. And highly recommend taking a read at this of this commentary. And so they're calling for a moonshot level new agenda for working on equity. And I'll just pull out, I think it's just worth reading, but I'll just pull out a couple of highlights. So pointing out that the process of how we conduct research is just as important as the results of research. I agree with that. And kind of like recalibrating how we're doing things, and not just sort of not an ends justify the means kind of outcome is a great view. And then, I think following on Joe Graves and sort of his liberal arts approach to science and to history and to writing, they've got a great quote in here from James Baldwin. Not everything that is faced can be changed, but nothing can be changed until it is faced. And I think that really frames nicely the situation science finds itself in, where we can't just sort of kind of pay attention for this for a few minutes and then let go. We really have to face it. And so this commentary has some really practical ideas and sort of simply arranged. So near the end, they go into bullet points, a little maybe even echo of when we were talking with Joe Graves. They did their when his book Racism Not Race, frequently asked questions with Q&A. Here it's bullet points, under different categories. What can you do in general? Here are 3 bullet points. How about for education? 3 bullet points. Scientific work culture, about 10 bullet points. Scientific progress and translation, more bullet points. And I think, really nice way of summarizing and just sort of thinking about, even in our daily scientific lives, how we might contribute to a more equitable research environment. And so again, fun to hear from Joe Graves and hear his voice on this important topic. So that's my pick of the week.
It's really good. Yep. His book is great too. I really enjoy that.
Yeah, me too.
But it's not easy. It's not going to be easy, unfortunately.
Exactly.
This pervades all of society, not just science, unfortunately, and it's very difficult.
Yep. How about you, Vincent? What's your science pick of the week?
So I have a wonderful resource from Fred Murphy that everybody needs to know about. It's a website called Foundations of Virology. And Fred Murphy is a retired virologist who Rich Condit and I interviewed a few years ago on TWiEVO. And he has written a book called The Foundations of Virology, which is a history of virology. He actually had a copy of it printed in his home. Rich and I went to his home to interview him. It was about 6 inches thick. Wow. It's huge, but you can get it here for free. The whole book is available. It's an incredible history of virology. And accompanying it, also on this website, he has electron micrographs of viruses. He has PowerPoint slide sets based on the book. He has a section called the Virology Papers. It's a Word document with a chronological table matching the PowerPoint slides and the book, a historical series of events in virology. It has a link to TWiEVO. There's a link to an interview before masks, the last interview before COVID came to occupy all TWiEVO sessions.
That's right. Wow.
And then another set of images for the study of the history of yellow fever. This is... Yes, go ahead. Sorry.
Oh, sorry to interrupt. Am I remembering right? Is it Fred Murphy who took that iconic electron micrograph of Ebola?
He did. And in fact, Fred sent me some of the original TIFFs, very high resolution, and I'm having some printed to put next to the wall. Right behind me is the painting by David Molesky. Of course, he was inspired by Fred's electron micrograph, which is iconic. And that's when I saw the painting, I said, "Oh my gosh, this is Fred's EM," right? So Fred has been sending us a lot of material, but they're all here on this website, Foundations of Virology, which is maintained at the University of Texas Medical Branch, Galveston. I mean, if you're looking for historical material on viruses, you have to go and cool. Check this out. I actually have been interacting with Fred You know what wall text is in a museum? There's a little card next to a painting. It tells you what it is, the artist. So I had that made for David's Ebola. And then I said to Fred, I want to put your EM up and I want wall text next to that. So he sent me the story of the day he took that electron micrograph. Here, listen to this. This is very cool. So there's an outbreak of Ebola hemorrhagic fever, 1976, right? This is the first one. There was previously an outbreak of Marburg disease hemorrhagic fever in Germany in a primate center. And the first time they see these filamentous viruses, which they'd never seen before, and the monkeys have hemorrhagic fever. So they ship samples from this African outbreak to various labs, and the CDC gets samples. The lady gets the package. She opens it up. All the vials are broken.
Jeez.
All of them. And Fred says anybody else would have thrown it in the autoclave immediately, but she took out a little piece of gauze, and she squeezed a drop of fluid out of it. She went in the lab and put it on Virocells, and 2 days later they were wiped out.
Oh, man.
And so she gave Fred a few microliters of the supernatant. He said he went into the EM and he took a picture without fixing it. So normally you would fix a sample, but he says that screws up the morphology. And so he didn't want to fix it. He took a picture and he said my heart sank. He saw these filamentous particles. He said, oh my God, this is Marburg virus. He ran into the office of the CDC director. He was holding this print. He said it was dripping from the developer. And he showed it to this guy, and they all realized it's a filovirus. And then a few days later, they did the experiments to show it wasn't Marburg. It was a new virus, and they called it Ebola virus. I just think, so I had that story put on the wall text, which I think is just amazing. And Fred actually talked about it on his TWiEVO as well. Isn't that cool? Isn't that a great
really
story?
Cool. Yep, kind of the history of it, and
it's Fred. Fred did that, and he said he printed it out with wet negatives, which is not great, but he wanted to do it really fast. This is the days when we had to take negatives and print them, which I remember very well.
Yep, yeah, and maybe it's this transition. So now, how do we identify these things with their genomes? Right, we sequence it, but here, how do you identify it with the image? You do the EM and you immediately say that looks like Marburg, and that connected it, right? And then the sequencing came next. Yeah, it was harder to sequence.
Well, the same SARS-CoV-2, the first images said, "Hey, this is a coronavirus," and then they sequenced it and said, "Yeah, this is a corona." Because the EMs can be very clear about what sort of virus, unless it's a brand new one that you haven't seen before, right?
Yeah, no, and remembering that the visualization, how important that is to virology. We talk so much about the genomes these days, but it's really that seeing is believing. Yeah,
for sure.
Very cool. Oh, and it looks like we have a listener pick as well.
You should take that now.
This looks great. I previewed this, and I have to say I can't get enough of this. So Sam writes with a listener pick, suggesting perhaps that I have a new hobby here. So he says, "Dear TWiEVO, I wanted to submit this cartoon in view of Nels's erstwhile pandemic beard. Might this show what Nels gets up to when he's not podcasting or generating cool science?" And so this is a great cartoon. We'll post the link. This is from Saturday morning breakfast cereal with Zach Wienersmith. And so I don't know, I'm not going to read through the whole thing here, but it's it's a young kid in bed who's scared about some monster under his bed. And so his dad comes. It turns out it's just an evolutionary biologist with a massive beard rambling about some sort of, points that are totally academic and, don't really matter for real life. And it works. The kid falls asleep almost immediately listening to this bearded evolutionary biology monster under the bed rambling. And the father of the young kid says, he went out like a light. So this is great. Thanks, Sam.
It looks like you, Nels.
Totally. Yeah, I'm starting to get the gray here. This guy's maybe a few years ahead, but I'm on the way either to Santa Claus or a evolutionary biologist who just drones on about academic points. We'll see. I might, for the next podcast, it might be in a different situation. I'm thinking about coming out of pandemic hibernation and really kind of cleaning up my act. So stay tuned. on that one.
I put the link to this cartoon in the chat, folks, if you want to check it out. It's very funny.
Yeah, that's funny. That's
Nels
a great one. Thanks, Sam.
hiding under the bed there. All right. That is TWiEVO number 76. You can find the show notes at microbe.tv/TWiEVO. If you want to send in a comment, a question, a pick, TWiEVO at microbe.tv. If you like what we do, consider supporting us. You go over to microbe.tv/contribute. A couple of ways you can contribute to the cause of science education. And since we are now a 501(c)(3), a nonprofit, your contributions are tax deductible. Nels Eldy's at cellvolution.org on Twitter. He's L Early Bird. Thanks, Nels.
Thank you, Vincent. Fun to be together again. Looking forward to the next one.
I'm Vincent Racaniello. You can find me at virology.ws. Music on TWiEVO is by Trampled by Turtles. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, stay curious. Thanks, everybody, for coming, and those of you in the chat, take care. Good to see you. Bye-bye.